Okay. Good morning, Ms. Sarah. Today we're presenting our project on the gut microbiome analysis of people suffering from ASD versus the healthy. This interactive app was built using Streamlit and explores the differences in gut microbial abundance, as well as the diversity between the individuals with ASD and the healthy. We'll guide you through the app's features, functionality, as well as its relevance to research on microbiology.
So our project is designed to simplify microbiome analysis by allowing users to interact with microbial data, apply filters, and visualize the results. Let's start by discussing how the app works and what its section does.
Okay, so let's start with the title, I guess, and the description of the app. So as you can see here at the top of the app, we have the title as well as like a introductory, like about this app and what we made and I guess like how we made it as well. So this is just to make everything clear and concise.
Okay, so next up we have how to use and why studying the gut microbiome matters for individuals with ASD. So this sets the stage for users, giving them a concise overview of what they can expect and why the data matters. Now let's move on to the first interactive session.
Okay, so for the sidebar controls here, we have like the minimum fold change. Like we can always change this the left part here from like 1 or 2 and all the way up till 10, as well as the top taxa from like 1 up till 25.
Okay, so basically what this filters does is it allows user to focus on more significant microbial differences, providing a customized view of data. Let's demonstrate this by adjusting the sliders to filter the data for taxa with a fold change of at least 2 and viewing the top 5 taxa pairs.
Okay, minimum fold change, let's set it to 2. And then the number of top taxa into pairs would be 5. So as you can see here, like it changes from the 1 shown here. And then once we put the slider up to like 5, and then it'll show 5 now. And it goes all the way up to like 25, like as we scroll all the way down. But let's just, for the sake of simplicity, let's just keep it at 5 right now. Also below the table, we have something called bacterial insights, which shows like the effects of the filtered data from the table here from the taxons and like basically here let's say bacteriotides, Stichoris, it's part of the normal gut flora, but overgrowth can lead to inflammation and it affects the gut, and it has more values in ASD compared to healthy. And this is all shown for every single taxon in this filtered data table.
Yeah, so basically this gives us like insight of the precompiled from the microbiome, and it just shows us what it does and everything.
Okay, next we have like for the visualizations, we have the heat map of the selected taxa, as well as the fold change for the selected taxa. So for the first visualization, we have a heat map, and this is like linked to this filtered data table right here. So basically how it works is if you put the number of top taxa in the filter here as like 5, as we have it right now, then here in the heat map, I'll also show like the taxon. So like let's start with like 1, and it'll be like this. And then once you like slide it all the way up, and then it can go up till, of course, like the 25th taxons. But again, let's just put it at like 5 for now.
Okay, so basically what this heatmap does is it shows the mean abundance of the selected taxa in ASD and the healthy groups. It's color-coded to make patterns and differences easy to spot. For instance, darker color means it's it's lower, as you can see here. And for this one, the yellow one, it means it's the highest one, and it's found in the ASD mean. And as you can see, the colors for the ASD means are like the ones on the higher ups, the more mean abundance, while the healthy mean is more of the darker color, which has lower mean abundance.
Okay, so next up on the list, again for the visualization tool, this is more like a bar plot, which highlights the fold change for the selected taxa. So in this case, uh, these are all the selected taxa that we have. And I guess this visualization shows like how much more abundant in terms of like multiplicity of the ones in ASD versus healthy. And it's useful to like identify potential biomarkers of like how the ASD or how the bacteria can cause people to have problems in ASD versus healthy.
Okay, so next up is one of our biolog bio
Yeah, computational.
computational biology measures is a Shannon diversity index distribution table. And this is a metric used to quantify the diversity of microbial communities, and our app calculates and compares the Shannon index for ASD and healthy groups.
So to visualize the data, we've included like a box plot showing the distribution of the Shannon index for both groups being ASD and healthy. So below this, we also actually put like the p-value that we got from the Mann-Whitney U test. And here the result that we got from the calculation is like 0.00443. But basically, since the lower the value of this p-value from the test means that there is a significant like cause and effect between ASD and healthy in terms of like the bacteria. So basically, there's a big difference between the ASD and the healthy, basically.
Okay, so to wrap up our app, to wrap up, our app is a powerful tool for exploring and visualizing gut microbiome data, where it combines interactive controls, dynamic tables, bacterial insights, and visualization to provide a comprehensive analysis of microbial differences between ASD and healthy individuals. By making this information accessible, we hope to contribute to microbiome research and help identify potential biomarkers for ASD. And you want to show the code?
Okay, let's move on to like the code for now. So of course, at the top here we have like the necessary imports and all that stuff. And then loading up the data set, and of course it's just like cleaning up and preparing the data and stuff. You wanna continue?
Yeah, it's just the pre-compute columns. So for the columns, of course, we're going to separate the ASD and the healthy columns. For the ASD one, we start everything with A, and for the healthy ones, we start everything with a B. Next time, we have the calculate mean abundance. Of course, for mean, we cannot calculate both ASD and healthy one. We have to make it a separate measure. So we have ASD mean and the healthy mean. Same thing also for the fold change and the Shannon index calculation function. And continue.
Okay, again, just of course after the Shannon index, we also have like the function like the ones that we shown before, where we show like the bacterial insights of like what the bacteria does in a human body, its effects and stuff like that, and where it affects, and like the amount of the bacteria with how much more it is found in. Is it like in ASD, or if it's found more in healthy? And of course, we picked the one like this more found more in ASD to show like more of a correlation. So here we have a couple of them, like the top ones. But aside from that, this is just like the Streamlit apps and the again like just brief description of the project and stuff like that.
Okay, so and of course, after that, we're gonna make the sidebars, which is so it's more interactive, making our app more interactive for the users. This filter parameters, as you can see previously from our app, is the sidebar, and you can change the full change. Then the top taxa. Oops, okay, and display filter data in Streamlit, and display bacterial insight. Basically, is the filter data is the table just now, and the bacterial insight to make it easier is this one, the filter data, bacterial insight. You just have to show it, and some heat map visualization, bar plot visualization, the Shannon index distribution, and the p-value. And that's pretty much it from our code and our app.
If you have any more questions, please don't hesitate to contact us through maybe email or WhatsApp, and stay tuned for the report as well, because that's where we'll be putting more information regarding this whole thing, including like the mathematical operations and all that good stuff. So yeah, thank you.
Thank you.